So this is GATE 2022 Life Sciences, Section P Biochemistry, question number 41. Which of the following carbohydrates have beta-1,4 glycosidic linkage? Okay, and the option are cellulose, B chitin, C lactose and D maltose. This is the NPTEL online course reference for this question. So first we'll understand what is this beta-1,4 glycosidic linkages. Okay, so here we have a glucose molecule. Okay, this is Fischer projection, right? But as you know, glucose is not found in the Fischer projection, but it is found in the cyclic position. Okay, right? And how this cyclic position of glucose is found? When this OH, the C5OH, when it attacks on the C1 carbon, right, we have a cyclic structure and we can have a pyranose. Glucose is a 6-membered ring, so we have a pyranose. Okay, so this pyranose can be of there are two isomers of this pyramers, alpha and beta. Okay, always remember this. In alpha we have OH below, okay, OH below the plane of axis, whereas in beta we have OH above. So this is how I remember this. So alpha, we are going down, right? So to write alpha, so this is how I write alpha. I go down, right, in alpha. So this is how I remember that in alpha the OH lies below the plane of this axis, right? Whereas in beta, for beta we write like this. This is how I remember. It might be useful for you also. So beta we go up. So the OH is above the plane of axis. So this is how I remember the structure, right? And these are the stereoisomers of glucose, right? What is stereoisomers? The stereoisomers are when the H and OH they differ in the plane, right? So OH here is below the plane of axis, whereas in beta it is above the plane of axis. So these are the stereoisomer of glucose. The molecular weight is same, right? So these are the isomers, but these are the completely two different structure of glucose. You cannot say that alpha D glucopyranose and beta D glucopyranose are same, right? These are two completely different structures, and our body recognizes as these two structures as completely different, right? So this is beta D glucose. So in the question they have asked, they want beta-1,4 linkage. So when this beta glucopyranose, it will have a linkage with, this is beta-1,4, that is it will have linkage with the 4th carbon of another sugar, right? Suppose there is this one sugar here. So this beta-1,4 linkage will have 1,4 bond, 1 carbon of beta glucose and the 4th carbon of the other sugar. This is the beta-1,4 linkage. So carbohydrates, also known as polysaccharides, they can be either homopolysaccharides or heteropolysaccharides, right? Very simple. As the name suggests, homopolysaccharides means the basic unit is same. So if the basic unit is glucose, cellulose, starch, glycogen, all these are homopolysaccharides of glucose only. But these are very different from each other, right? Starch is very different, glycogen is very different, cellulose are very different. But the unit is glucose. All those three polysaccharides have glucose as their basic unit. Okay? And what is the difference? I'll tell you. So polysaccharides. What are polysaccharides? The basic unit, the length varies from at least So the glucose units in starch, glycogen, there are thousands of glucose units. So those are polysaccharides, right? Where the glucose units can be more than, so there is a range. So for polysaccharide, I think the glucose unit should be more than 10. Okay, then it is termed as polysaccharide. Disaccharide, which means it has only two basic sugars, simple sugars. Okay, we also have heteropolysaccharides, and so heteropolysaccharides, again we have branched and unbranched. In homo also we have unbranched and branched. Okay, so always remember that so always remember, branching always occur. Generally it's not a thumb rule, and it's not generally we see that branching occurs at 1,6 position. Okay, so this is where the branching occurs. The one carbon will attack the sixth carbon, and this is how the branching occurs. And generally this unbranching of unbranched polymers, the unbranched polymers, they form different units through 1,4, right? 1,4. So here we'll have a one unit, it will form with the C4. So here we'll have, it will form with fourth carbon. So the branching always occurs at 1,6, and the straight chain formation of unbranched units occurs at 1,4. Okay, so what is the difference? So as I mentioned earlier, cellulose, starch and glycogen, these are completely different from each other, right? We in humans we have glycogen. In plants, what do we? Both cellulose and starch. So cellulose is a unbranched. Okay, cellulose is a unbranched homopolysaccharide. It has no beta, or it has no 1,6 linkage. Okay, this sixth carbon is not linked to, there is no branching in the sixth carbon. So cellulose is an example of a homopolysaccharide which is unbranched, whereas starch and glycogen is an example of a branched homopolysaccharide. They have a branching. So both starch and glycogen has a branching. So how does they differ from each other, starch and glycogen? Okay, the major difference between cellulose, starch and glycogen is, in cellulose we have beta 1,4 linkage. Okay, whereas in starch and glycogen we have alpha 1,4 linkage. Okay, so we are not able to digest cellulose. Okay, if we eat plant leaves, we won't be able to digest it. Why? Because it has higher contents of cellulose, and our body cannot break the bond. This beta 1,4 linked glucose units, these are the basic units of cellulose, right? Cellulose is an unbranched beta 1,4 linked D-glucose unit. Our body doesn't break the beta 1,4 linkage. Okay, that's why we eat cellulose as a fiber, okay, to increase the fiber in our diet, but it is not digested by our body, right? So the biggest difference between cellulose, glycogen and starch is we have beta 1,4 linked glucose, and in glycogen and starch we have alpha 1,4 and alpha 1,6 linkages. So starch and glycogen both have alpha 1,4 linkage. So what is the difference between starch and glycogen then? We are able to eat starch, right? Our body is able to digest starch. So potato is starch. The french fries which we eat is starch. We are able to digest the starch. The sweet potato in our, it is daily, it is given in our diet daily. We eat it, we digest it. So starch and glycogen are the same chemical structures. There is one striking difference between starch and glycogen, and that is glycogen has various branching. Okay, so in starch, as you can see, starch, this is amylose, a blue chain, right? This is amylose, and we have a branching here. So in starch, at every 5 to 6 residues there is a branching, and that branching chain is known as amylopectin. But in glycogen, the most important thing is there is branching at every unit, at every other possible unit. So glycogen is highly branched, okay, as compared to that of starch. The only difference between starch and glycogen is the glycogen is highly branched, right? Glycogen is a storage carbohydrate in our body. It is highly branched. Okay, that is the only difference between glycogen and starch. And cellulose, so it is, first of all, it is unbranched, and we have beta 1,4 linkage. Okay, this is very important. Why we are not able to digest cellulose? Why we are not able to digest the grass? Cows can digest the grass. We are not able to digest the grass because it has beta 1,4 linkage. Our body is incapable of breaking down beta 1,4 linkage between the glucose residues. Okay, that's why we are not able, we won't be able to eat. Obviously we can eat grass, but our body will not digest it. The next is next we had chitin. Okay, so chitin here, chitin is also a homopolysaccharide, right? So chitin is found as an exoskeleton in many insects like beetle and cockroaches. N-acetyl glucosamine. So here this is N-acetylated glucosamine structure. Okay, at every second carbon we have acetylated N-acetylated amine, right? So the basic unit of chitin is N-acetyl glucosamine. It is a homopolymer, right? It is a homopolymer. This is also not found in human. Chitin is not found in humans. And so which is this linkage? Tell me now, this is which linkage? So this is beta 1,4. We have to check the beta link of from the first C1 carbon. Okay, this is very simple to observe. This is beta, right? The hydroxyl group of beta is above, right? So this is beta 1,4 linked, right? So we have cellulose which has beta 1,4. Again, the trick is chitin is not found in humans. So any so beta 1,4, it is, we have difficulty in digesting. Because we are not able to cleave this. We are not able to cleave glucose beta 1,4 linkage. Also N-acetyl glucosamine is not found generally in, it is found, but beta 1,4 linkage of N-acetyl glucosamine we are not able to cleave, and these are found in insects. Okay, so in chitin also we have beta 1,4 linkage. In cellulose also we have beta 1,4 linkage. In glycogen and starch we have alpha 1,4. And then next we had option was maltose. Okay, so this maltose here, the name is already given. So the OH is below, right? If the OH is below, it is alpha, right? If the OH is below, it is alpha. So maltose is what? It is a disaccharide of 2 glucose subunit. So first subunit is alpha glucose, and here this is beta glucose, correct? This is beta. So the first carbon here, the first carbon is attacking to the fourth carbon to get alpha D glucopyranose 1,4 D glucopyranose. Okay, so this is beta, but this is alpha 1,4 linked. Right? Why alpha 1,4 linked? Because the C1 is attacking C4. Right? So this linkage is alpha 1,4 linkage. We will not call it beta. Which is this sugar? Lactose. Lactose is also a disaccharide. Okay, lactose is also a disaccharide, and two basic units of disaccharide of lactose are galactose and glucose. Okay, so this galactose is also beta and this glucose is 1,4. We are able to break lactose. Why? Because this is galactose 1,4 beta galactose 1,4 pyranosyl. Right, what I said earlier, beta glucose 1,4 linkages cannot be broken by our body, but beta galactopyranosyl 1,4 linkage can be easily broken. Okay, there are few people who are not able to digest, who are not able to break down this lactose. Those are lactose intolerant people, right? They have some modification in their enzymes. They are not able to digest this beta 1,4 linkage. Okay, so this gal beta 1,4 linked glucose. Okay, so this is one of the important table in the lineages. So starch, as I mentioned earlier, it's a homopolymer, right? It's a homopolymer of alpha 1,4 linked, right? A linear polymer is known as amylose, whereas a branched with a linear branching at 6th glucose, right? It's known as amylopectin C. So the branching occurs at 24 to 30 residues in starch. And starch is an energy storage in plants. Whereas glycogen is also homopolysaccharides. It has 1,4 glucose, right? Same as starch, no difference. Alpha 1,6 same as starch, no difference. And the branching occurs at every 8 to 12 residues, right? In glycogen there is excessive branching, right? And glycogen is a energy storage in bacteria and animal cells. Humans store glycogen, whereas plants store starch, right? Wheat, potato, that is the stored starch in plants. Whereas cellulose is a homopolysaccharide with beta 1,4 linked. It is unbranched, right? So there is no 1,6 branching. Cellulose is a homopolysaccharide. Also chitin is a homopolysaccharide. It has beta N-acetyl glucosamine, right? It is found in insects, spiders, cockroaches. It is the exoskeleton of these insects is made up of chitin. Then we have dextrin. Dextrin is also homopolysaccharide. It has alpha 1,6 linkages, and see the branching here occurs at 1,3. Okay, so now till we have seen the branching occurs only at 1,6 in starch and glycogen, but in dextrin the difference is it's a homopolysaccharide of glucose only. It has basic unit of glucose, but the branching occurs at 1,3. Okay, and the single chain without any branch, it is 1,6 and not 1,4. Okay, this is the only difference between dextrin and our starch glycogen cellulose homopolysaccharide. Okay, it is found in bacteria and as an extracellular adhesive. Then we have peptidoglycan. Okay, we all know peptidoglycan. It is found in bacteria, cell wall of bacteria. So peptidoglycan is a mixture of carbohydrate and peptides. It is a heteropolysaccharide first of all, and it has peptides also attached. So it has N-acetylglucosamine, right? And to this N-acetyl linkage we have different peptides, either N-linked or O-linked. Then we have agarose. Agarose is also a heteropolysaccharide. It is made up of galactose beta 1,4 and 3,6 anhydrogalactose. Okay, agarose is found in various algae and it is also a cell wall material of various organisms. Then we have a hyaluron. Hyaluron is found in humans also, right? It has acidic residues. So hyaluron is a hetero. It is made up of an N-acetylglucosamine. This is acetylated glucose, right? This is acetylated glucose and N-acetylglucosamine. It is found in invertebrates, extracellular matrix of skin, connective tissue. It helps in viscosity and lubrication in joints. Okay, so beta 1,4 linkages is found in cellulose. It is found in chitin, right? Maltose is alpha 1,4 linked. Galactose is beta 1,4 gal. It is not glucose. Okay, so the answer is A and B.